So yeah, thanks, Joanna, for the introduction. As Joanna said, my name is Scotty Blechman, and today I'm going to be talking a little bit about Scilligence's Mol2HELM tool in RegMol. So Scilligence has always kind of been an industry leader in the area of Helm support, and I think we're very proud to kind of present this tool that helps companies that may still be using old legacy systems adopt Helm format. So I do want to start with just a brief introduction to Helm. I'm sure many of you are already familiar with Helm, but just to kind of get everyone on the same page, I want to briefly go over exactly what Helm notation is and why it's needed. I'm going to talk a little bit about the Mol2HELM tool developed by Scilligence. So this is not a brand new tool, but it is something that we've done a lot of active development on recently and are looking to continue doing active development on. And I'll talk a little bit about why such a tool is needed. I'm also going to go over a case study of a client we worked with who had used Mol2HELM as a kind of major player in their data migration. And then lastly, I want to talk about kind of future directions for where we want to go with the tool. So starting off with a brief introduction to Helm, like I said, as Alan mentioned, Helm is the hierarchical editing language for macromolecules. So what do we mean by hierarchical? So the hierarchy starts at the atom bond level. In this example, I've shown glutamic acid or glutamate. So the atom bond structure is going to tell you what atoms are connected to one another, what bonds connect them. But what it doesn't tell you is how can this structure be used in an actual biopolymer. So the atom bond level is only giving you the information about the monomer structure. So if we go up to the monomer level, this is a screenshot from Scilligence's monomer manager inside RegMol. And what this does is it lets you define how exactly that atom bond structure is going to be used in an actual polymer. For example, I have glutamic acid defined here, and I've defined different R groups or leaving groups where the other monomers in the polymer are going to connect to this monomer, as well as some other miscellaneous metadata such as natural analog and polymer type. What's missing here, though, is how does this monomer now fit into the whole sequence? What's the whole sequence, not just this one monomer? And so then we move up to the simple polymer level. So here I have an example that is just a short linear peptide. On top is the atom bond structure. And then just below that is the pictorial representation of the Helm, followed by the Helm string and the sequence. And so in a case like this, it may appear that Helm is just adding this unnecessary extra text to your sequence because it's something you can kind of interpret relatively straightforward when you are given the sequence. But where Helm really starts to shine and where we really start to see the need for Helm is when we get to more complex polymers. So this is one example. I've taken the peptide from the previous slide and just cyclized it. And this is not even a particularly complex case. I've added one piece of complication, just cyclizing the peptide. And yet, if I just try to call this the cyclic version of that sequence, I've already introduced multiple questions. Which monomers are connected in the cyclization? Are the monomers connected from N terminus to C terminus, or are they connected by their side chains? There's a lot of information that you can't interpret in just plain text. And so this is where Helm really shines by explicitly defining for you in the Helm string which connection points of which monomers are connected to one another in a machine-readable format that can be processed by automation and AI tools, as well as by Scilligence RegMol. So here's just one more example of another more complex polymer. In this case, I've connected my cyclic peptide to a single-stranded oligonucleotide using a PEG2 linker, which is called a chem in Helm. And again, you can see the Helm string is explicitly defining for you what your connection points are, which monomers are connected to one another, information that would be very difficult to just describe verbally. So that's HELM. What I want to talk about next is Mol2HELM, which is, like Joanna mentioned, a tool that Scilligence has developed to help companies adopt HELM notation as well as for day-to-day use. So this is what the Mol2HELM UI looks like. You can enter your atom bond structure, and just with the click of a button, it is converted to our pictorial HELM representation. You can also copy the HELM string out of there, or even open up the HELM web editor, which we developed. And we are also able to do auto pairing here. So you can see the small molecule representation does not have any pairing information. And when I've converted to HELM notation, I've been able to just use the Scilligence auto pairing tool to auto pair even with some non-conventional nucleotides like inosine. So what is Mol2HELM exactly? Mol2HELM is a molfile to HELM conversion tool. It's made to make it easier to take just an atom bond structure and identify exactly what the HELM string should look like accordingly. Reverse conversion, of course, is also available, HELM to mol. That is very much the easier part. You have your HELM string, you look at your monomer definitions, and you expand them out. But the real challenge of conversion between HELM and molfile is in this direction, Mol2HELM. So our Mol2HELM tool uses algorithms to cut the molecule into fragments at certain points and identify them based on the defined monomer library in Scilligence's monomer manager. So this is really the biggest challenge that Mol2HELM aims to address, is determining in an automated way which kind of bonds of the molecule to cut to get your individual monomers and figure out your connections. Mol2HELM currently supports both oligonucleotide and peptide structures. Originally, it was just for peptides, and we've since expanded support to oligonucleotides. In the future, we're also hoping to support peptide oligoconjugates as well as chem monomers, which are kind of chemical modifiers that don't fit into either category. And we also have added support in recent years for duplex oligonucleotides. As for inputting structures into Mol2HELM, we like to keep it flexible. You can free draw an atom bond structure in the Mol2HELM box, as I showed in the UI on the previous slide. You can also upload input structures, mol files, smiles, INCHI, ChemDraw files, anything that is supported by our JS Draw Sketcher is supported by Mol2HELM. And possibly even more importantly, we have RESTful API calls available to Mol2HELM. We are seeing more and more in recent years that people are preferring to interact with these types of tools via API as opposed to just through the UI. And so we've made sure that everything that works through the UI for Mol2HELM is also functional through our RESTful API calls. So why did we develop Mol2HELM? I touched a little bit on this, but HELM is a relatively young and evolving technology, but it's increasingly becoming a standard. So HELM, I believe, was started in 2008, and it is now kind of the standard notation for oligonucleotides and peptides. It's been used in a lot of different publications, and it's used across a lot of organizations. And it has the potential to replace dozens of legacy formats that lack interoperability. So lots of companies prior to HELM notation had their own legacy formats that just did not translate between one another, that were only understandable to people within the organization, and it just failed to kind of foster collaboration. So HELM is kind of looking to address that challenge, and we want to help people adopt HELM to address that challenge. A lot of older software tools don't have HELM support, and so a lot of clients may have been working with molfiles or other notations prior to moving to Scilligence Regmol. And so this is a way of helping people get on board with Regmol and HELM. Legacy data sets are also often in molfile format, again, because older software tools often don't have HELM support. And we also have the case where clients may work with molfile on a day-to-day basis, but need to generate HELM for registration in RegMol or for their publications. So people may work with molfile day-to-day because they need to see the atom bond structure, they need to look at individual sections of the molecule, but when they are going to register or when they're writing up a publication, they don't want to publish the entire structure expanded because it's more information than people need. Additionally, Mol2HELM has the potential to allow for easier structure sharing within organizations. If you imagine a very large biopolymer, you don't need to expand the entire molfile and send that to your colleague. You can condense it into HELM format using our Mol2HELM tool and just send the HELM string to your colleague, who can then very easily do that reverse conversion. So I also want to discuss a case study. This was a project that I personally did a lot of work on, helping one of our clients kind of get on board with HELM by helping them with a legacy data migration. So in this project, the client had oligonucleotide and peptide legacy data. They had thousands of oligonucleotides and peptides, all in molfile format with no sequence information. These oligonucleotides and peptides were made up of both natural and non-natural monomers, and there was no base pairing information present on the duplex oligonucleotides. So this kind of presented a unique challenge that Mol2HELM was equipped to handle. The client wanted to adopt RegMol because of our strong HELM support. The previous registration system they were using did not have HELM support, and registering these structures in HELM format would allow for them to be more readable through the pictorial representation, be more searchable by searching by what monomers are present in the structure instead of having to do small molecule substructure searching, as well as organizing their data, again, based on what monomers are present and how they're connected, instead of looking at every atom and bond. Additionally, their duplex oligonucleotides needed to be auto-paired upon conversion to HELM. And that was also something that we were able to help with. So in this particular case, we helped the client first import their monomer library into RegMol. This was a relatively straightforward task because our monomer manager supports upload of SD files or JSON. And so we were able to just get the monomer definitions from the client and upload those into our monomer manager. After that, we wrote a script to process their mol files and send them to Mol2HELM via the RESTful API call that I mentioned. This was done in an automated way such that we could just start the script and leave it running until it was done processing the data. And what we found was that for over 95% of the records in the client's legacy data, we had seamless conversion to HELM notation. So all of the parts of each structure were identified as monomers and identified, more importantly, as the correct monomers. As for the remaining records, HELM has support for something called inline smiles, which is essentially a way of allowing you to include sections of the structure that you don't necessarily want to define as a monomer, sections that are kind of meant to be free drawn because they're only ever going to be used once. And so the way Mol2HELM handles fragments that it can't identify as monomers is it converts them to inline smiles. So because we were able to get that conversion from Mol2HELM with the inline smiles, we were actually able to analyze this small subset of less than 5% of the records to determine for each record which cases were kind of meant to have inline smiles, were meant to have free-drawn portions of the structure, as well as, even more importantly, which cases were kind of structures that we saw repeatedly used across several of these records that were kind of missing from the monomer library. So having this kind of small subset that had partial conversion allowed us to look at that small subset, which is much more manageable than looking at the entire data set of thousands of structures, and kind of figure out which cases were kind of gaps in Mol2HELM functionality, which cases were missing monomers in the client's library, and which cases were intentionally inline smiles. So as for our future plans for Mol2HELM development, as my colleague Alan mentioned, conjugates are a huge up and coming kind of player in recent years in the pharmaceutical world. And so definitely our top priority is adding support for conjugates. In particular, peptide oligoconjugates will be relatively straightforward for us to add support for. We also want to look into adding support for peptide chem conjugates, oligochem conjugates, as well as peptide oligochem conjugates. So cases like the one I showed earlier, where you have a chem linker between your peptide and your oligonucleotide. Additionally, we want to support kind of more complex and rare cases. So things like having cyclization between 3 different monomers via a linker, where your linker is connected to each of those 3 monomers, kind of holding the whole structure together. We're looking to support hairpin oligonucleotides, where the kind of middle of the sequence wraps around and forms a hairpin, as well as unconventional oligonucleotide backbones, for example, glycol nucleic acid, which is kind of drastically different from a ribose backbone, and thus needs to be identified differently in the algorithm. So that is all I have for today. Thank you for listening, and yeah, I will take any questions, or I guess I can look at the Q&A. So Scott Starry asks, does V3000 convert to HELM with the Mol2HELM tool? Not sure how this would be represented in HELM. So yes, so in Scilligence's monomer manager, you can define each monomer in molfile format. And so that includes support for V3000 because Scilligence Regmol supports V3000 molfile. So the V3000 molfile itself is not in the HELM string, but it's in the definition of the monomer. Frank asks, any interest in including HELM translators in Data Warrior? That is not something that we are currently looking into, but it's definitely a future direction we could go with the tool.